afternoon, ma'am. Good afternoon, everyone. I'm Lakshmi
And I'm Hemapriya.
Today we are doing presentation on nucleic acid. First part will be taken by Hemapriya and the further part will be handled by me.
In this topic, we are going to know about introduction, discovery and structure of purines and pyrimidines, nucleosides and nucleotides of DNA and RNA. Introduction. In the mid-19th century, the scientists began to develop the chemical composition of cells. It was young Swiss physician Friedrich Mister who in 1986 isolated a new substance from nuclei of white blood cells. As we all know that the white blood cells help us to heal. He called it mucilin, marking the very first discovery of what we now know as nucleic acid. This finding set the stage for understanding the molecular basis of heredity. Discovery. At first in 1969. At first in 1869, Friedrich Mister identified the mucilin in the cell mucilin, a substance which phosphorus from white blood cells. After Friedrich Mister, in 1889, Richard Altman.
Slides ಚೆನ್ನಾಗಿದೆ ಗಟ್ಟಿಯಾಗಿ ಮೇಲೆ ಮಾತಾಡು ರೆಕಾರ್ಡ್ ಆಗ್ತಿದೆ ಅಲ್ಲಿ.
In 1889, Richard Altman coined the term nucleic acid after determining its acidic properties and chemical composition. After Richard Altman, in 1944, Avery and MC Clarity demonstrated DNA.
Avery McCarthy
demonstrated DNA as the genetic carrier, transforming bacterial properties in groundbreaking experiments. After him, in 1953, Watson and Crick unveiled DNA's iconic double helix structure, a landmark moment in biology explaining its replication. In 1953 is known as the golden era of development of biological science.
Slides ಚೆನ್ನಾಗಿದೆ ಗಟ್ಟಿಯಾಗಿ ಮಾತಾಡಮ್ಮ.
Photo 51. The photo 51 is the famous X-ray diffraction image of DNA taken in 1992 by Rosalind Franklin and her student Raymond Gosling at King's College London. It provided critical evidences for the helical double helix structure of DNA. The X-shaped pattern in the image divided the DNA and DNA had the helix structure, regular spacing between the strands, a repeating pattern after every 3.4 nanometer. This photograph was later shown without Franklin's permission to James Watson and Francis Crick, who used it to build their DNA model in 1953. Without Franklin's knowledge, the image was shown by Maurice Wilkins to James Watson and Francis Crick, who immediately recognized its importance and used its data to define their double helix model of DNA. And it was published in 1953, while Watson, Crick and Wilkins later received the Nobel Prize in 1962. Franklin's contribution went largely unrecognized during her lifetime. Today, the photo 51 is celebrated as a turning point of the discovery in DNA structure and symbol of both scientific brilliance and the recognition of women in science. These are the some X-ray diffraction cameras. The first one is Eisenberg camera, second one is precision camera, and the third is X-ray diffractometer. And the fourth one is used by Rosalind 3.4 nanometers, and this is known as the minor groove, and the large one is known as the major groove. The outside diameter is 20 angstroms, that is 2.0 nanometers, with the cylindrical outline of the double helix are two grooves, the smaller one and the larger one. The minor size alongside the strands represent many negatively charged phosphate groups along the entire length of the each strand, and the further topics will be covered by Lakshmi.
Once again, good afternoon everyone. Thank you Hema for the introduction, and the further part will be handled by me. So Hema has already given some brief information about DNA. Now whatever topics I'm going to handle is not much complicated, so make it simple. So first is difference DNA and RNA. DNA stands for deoxyribonucleic acids and RNA stands for ribonucleic acids. Basically, it is known as deoxyribonucleic acid because it lacks one oxygen atom. So the word deoxyribonucleic acids, and RNA, that stands for ribonucleic acids. So DNA helps in genetic storage. As we know, DNA helps in genetic storage, which will be carried from generation to generation. So DNA is what we inherit from past generation. And it is basically a double helix structure wherein the RNA is a single stranded structure, and we can find DNA in nucleus and mitochondria, whereas RNA is found in cytoplasm and ribosomes. And DNA, the sugar found in DNA is deoxyribose, wherein the sugar found in RNA is ribose. Bases present in DNA are simultaneously. Let us learn about the difference between DNA and RNA. So the base pair present in DNA is ATGC, where in RNA it is AGUC. So can anybody tell what ATGC? AGUC? So the base pair which varies in DNA and RNA is thymine is present in DNA, wherein uracil is present in RNA. That's the base pair difference found in DNA and RNA. Where another difference, what you can write is, it is deoxyribose sugar which is present, and here we can find ribose sugar. So basically these two base pairs make a difference. So now we'll see what's the function of DNA and RNA. DNA is basically a very stable structure, and RNA is less stable. So can anybody tell what makes DNA a stable structure?
A double
Double stranded, and it is also less reactive. It is less reactive, which makes it more stable, wherein RNA, it has a presence of hydroxyl group. Basically, hydroxyl group is very reactive, and therefore RNA generally doesn't help in genetic storage. DNA does that job. So it is basically more stable, where this will be less stable, and it is less reactive, and RNA will be highly reactive. That's the difference between DNA and RNA. So the function, the structural difference contributes greater stability. That means the double helix structure is what it gives, it makes it a stable structure. And it also helps in making it ideal for long-term genetic information storage. Due to which the double helix structure and it being more stable, it helps in genetic information which will be inherited next generations. Wherein RNA is additional hydroxyl group. So here it lacks one oxygen atom, wherein in RNA you can find one more hydroxyl group, that is OH group. It renders it more chemically, it is less reactive as I said and less stable, suiting its role as a temporary messenger and catalyst. So now, what are types of RNA? mRNA stands for messenger RNA. RNA stands for rRNA stands for ribosomal RNA and for transcription. So you can basically remember it as MTR. You know, you all eat MTR brands. So you can remember it as MTR, and the DNA are aDNA, bDNA and zDNA. The DNA which is found in us is B form. So now we got to know the types of nucleic acids. Next we will know about purines and pyrimidines. It is something which makes, they are known as building blocks of nucleic acids. So purines and pyrimidines. Basically purines are double ring structure and pyrimidines are single ring structure. This question comes for 2 marks, so you can just draw the diagram structure of purines and structure of pyrimidines, and you can write this much information is enough for 2 marks. So you can write that basically purines are double ring structure and pyrimidines are single ring structure. The base pairs present in purines are adenine and guanine, where in pyrimidines it is cytosine, thymine and uracil. You can remember it as pyrimidines, you can remember it as cut. Here AG. Purine is basically a heterocyclic aromatic organic compound. It consisting of a pyrimidine ring fused to an imidazole ring. It is characterized by the larger size. Basically purines are larger in size than pyrimidines and they are double ring structure. The difference between purines and pyrimidines are their double ring structure and their single ring structure. This form the broader basis within DNA and RNA. As they are larger molecules, they form the broader basis within DNA and RNA molecules. And pyrimidine is a heterocyclic aromatic organic compound which is similar to benzene and pyridine, containing 2 nitrogen atoms at position 1 and 3 of the 6-member ring, defined by their smaller ring structure, which is crucial for precise base pairing within the helix. So this is the structure of pyrimidine, you can note down, and if asked you can write small definition that it is a heterocyclic aromatic compound, it is a 6-membered ring, and you can mention the base pairs present in pyrimidine, and you can draw the structure for 2 marks if asked for 2 marks. Of pyrimidine is C4H4N2. These are the number of molecules present in pyrimidine. That means they'll have 4 molecules of carbon and 4 molecules of hydrogen and 2 molecules of nitrogen.
Really, the 2 molecules nitrogen.
The structure should be. Sorry, that's the wrong structure. I write the structure here, sorry, you can copy this one. So structure of pyrimidine, and you can write the definition for 2 marks. The forms and important biological importance of pyrimidines is it includes nitrogenous base, that is cytosine, thymine and uracil. It is their essential components of DNA and RNA. So this is structure of purines. So the molecular formula of this is the structure of pyrimidine, and you can note down the structure of purine. So the general formula, the molecular formula of purine is C5H4N4, that it has four nitrogen molecules. And purine is the parent structure of the important biological bases. The nitrogen bases present again are adenine and guanine, which pair with thymine and cytosine. Always the adenine base pairs with thymine and guanine base pairs with cytosine. It plays a key role in storing and transmitting genetic information. So its function is to store and transmit genetic information. It is also part of several vital biomolecules such as ATP, GTP and coenzymes like NAD+. Nucleosides and nucleotides. So another building blocks for nucleic acids are nucleosides and nucleotides. Nucleoside as, so basically nucleoside as nitrogen base plus sugar, and nucleotides, it has nitrogen base plus sugar and a phosphate group. Phosphate group will be present. Nucleoside, it is a fundamental unit composed of a pentose sugar covalently bonded to a nitrogen base. Nucleotide and one or more phosphate groups are attached to a nucleoside, creating an activated building block. And between this there will be a chain formation, and the bond present between two molecules, bases between nucleotides and phosphodiester bond. So this is nucleotide. Nucleotide is the basic structural unit and building block of nucleic acids such as DNA and RNA. Each nucleotide is made up of three components, that is nitrogenous base. It can either be purine or pyrimidine. So nitrogenous bases are adenine, guanine, cytosine, thymine or uracil. So you can simplify and write it as it can either be purine or pyrimidines. And it has a 5-carbon sugar. That is, in DNA it is deoxyribose, and in RNA it is ribose sugar, and one or more phosphate groups will be present. The phosphate group is attached to the sugar of 5 carbon, wherein it is linked to the another carbon, that is 1' carbon, through a beta-N glycosidic bond. Nucleotides are essential not only for forming DNA and RNA strands but also for storing and transferring energy. This is because they have the presence of nitrogenous base which does this work, that is transferring energy in the cell, as seen in ATP, that is adenosine triphosphate. They also play roles in cellular signaling and metabolism. This is the structure. Nucleoside. A nucleoside is a chemical compound formed when a nitrogenous base, that is again either purine or pyrimidine, is attached to a 5-carbon sugar molecule. It can either be ribose in RNA or deoxyribose in DNA. Unlike a nucleoside does not contain phosphate group. So the difference is it contains phosphate group, wherein nucleotides contain phosphate group and nucleoside will not contain phosphate group. That's the major difference between nucleotides and nucleosides. Again, the same information, that is examples of nucleosides include adenosine, guanosine, cytidine, thymidine and uridine. Nucleosides are important because they serve as building blocks for nucleotides, which in turn make up DNA and RNA, playing key roles in storing and transferring genetic information. Chain formation. This is the chain formation. A nucleoside is a chemical compound formed when a nitrogenous base, that is purine or pyrimidine, is attached to a sugar molecule which can either be ribose or deoxyribose. Same information, that nucleotide and nucleoside will form a chain. Why nucleic acids matter is it contains genetic legacy and revolutionizing feats and in shaping life. So this is the last slide. You can note down important